Welcome to workshop. As an AI or ML engineer entering drug discovery, it is not always obvious where biological datasets actually come from or what happens to them before you see a file. This workshop is about building that intuition, tracing biological data from raw instrument output to model-ready files. So by the end of this workshop, you will understand how biological datasets evolve through multiple processing steps, which is different from pre-processed datasets you may have worked with in other domains. So you will learn how risk is introduced at each layer through noise, bias, and labeling inconsistencies that are specific to biological instruments and experiments. And you will leave with a concrete set of questions to ask when you receive the data from a scientist. And then so this gives you a structured pre-model checklist, and then also this workshop should give you a better sense of how to collaborate with scientists to validate assumptions you may not even realize you are making. So actually, in the last lecture, we traced how biological data are generated from a physical signal to a digital file. From a scientist's perspective, the key point is, by the time data reaches you, a lot has already happened to it. Before you write any code, you need to understand where the data originated and how it was transformed as it moved away from experiment. uh, so raw instrument data is filtered, normalized, and aggregated, and labeled so that it becomes useful. And each of those steps reflects scientific judgment. So those decisions determine what biological signal remains in the final file. So if they are not understood, models may end up learning patterns introduced by processing rather than the properties of the underlying biology that you are actually interested in. Now let's see this shared responsibility for data integrity. So one important thing to understand about working in drug discovery is that data integrity is a shared responsibility. In experiments, data is generated and first analyzed by scientists, and they understand what that instrument is measuring, how an assay should perform, and what sources of biological and technical variation are to be expected. That experimental analysis establishes whether a given data set is meaningful and interpretable on its own. So, whereas modeling, on the other hand, usually happens, downstream when data from many experiments, batches, or studies are brought together. That is where computational rigor becomes critical, ensuring consistency across data sets identifying technical artifacts, and making sure information can be combined in a meaningful way. So neither perspective is sufficient on its own. Reliable modeling depends on experimental understanding upstream and careful integration downstream with constant communication between scientists and engineers. So now let's see, where choices shape data. So how this transformation hierarchy happens. Let's talk about something that is a bit specific to biological data, the transformation hierarchy. I mean, we covered these concepts in the previous lecture. So if you are not sure about what I'm talking about, watch that first. Biological data exists in layers, moving progressively farther away from the original measurement. So at the bottom, you have a raw instrument output. Let's say FASTQ files from DNA sequencers, or raw TIFF images from microscopes, spectral data from mass spectrometers. So these files are large, noisy, and highly instrument specific. And then next comes process data. So that would be aligned sequencing reads, segmented cells, or peaks identified, for example. So this is where domain specific algorithms are applied, and the choice of algorithm matters a lot. So then there is interpreted data. So that could be annotated variants, or maybe classified cell types, identified metabolites. So this is where biological knowledge and assumptions begin to shape the data explicitly. Finally, you often work with the aggregated data, patient level summaries, gene expression readouts, or feature tables where individual measurements have been collapsed into higher level representations. Each step preserves some information and discards other information. So understanding what was kept, what was removed, and why is that's essential for making sense of the final file you receive. Let's start with an example, that is sequencing. So let's walk through this idea a little bit more clearly. Suppose you are working with genomic sequencing data and you receive a file listing genetic variants identified in patient samples, let's say. From a scientist's perspective, that file already reflects a long chain of experimental and computational decisions. One of the first questions is what reference was used to interpret the data. I mean, since variant locations and interpretations depend on the choice, you also need to understand how raw sequencing reads were aligned and how variants were identified. So there are a lot of methods. So different methods make different trade-offs, especially in low quality or ambiguous regions. So quality thresholds matter, right? Decisions about what counts as a reliable signal versus noise can dramatically change the final list of variants. Some regions of the genome may even be excluded entirely because measurements there are considered unreliable, for example. Finally, you need to ask about batch effects. Were samples processed together or across different runs or different instruments? Or what are the time points? So none of these questions are obvious if you are new to biological data, but each one shapes which variants appear in the final file and therefore what biological signal is available for downstream modeling. Okay, so now let's see another example that is from imaging. So, suppose you receive a CSV file containing extracted cell features, measurements like cell area, fluorescence intensity, or shape descriptions, for example. From a scientific perspective, that file already reflects many upstream decisions. The first question is how individual cells were identified in the images. Because different segmentation approaches handle overlapping or touching cells, a bit different. And those differences propagate directly into the feature values you see. You also need to understand how features were calculated and normalized. Choices about background correction, illumination, intensity normalization, all these things affect whether measurements are comparable across images or not. Another important question is which cells were excluded. Cells touching image borders, very small objects, or ambiguous regions, they are often filtered out. And those exclusions change the population your data represents. And finally, you need to know how the images were acquired. Same thing, were all images collected on the same microscope? Maybe is it the same magnification and settings? Or multiple instruments were used, or the conditions were any different? So you should be asking all these questions. Now let's see clinical data integrity. So now suppose you receive a patient cohort table containing lab values and clinical outcomes. One of the first questions is how patients were selected. Does the cohort include all eligible patients or only those with complete data? Things like that. And decisions like this introduce selection bias very early. You also need to understand how lab values were handled. Different hospitals use different reference ranges, for example, and normalization practices can be different by site, patient population, or even overtime outcomes. So definitions also matter. Clinical endpoints like disease progression or response are often defined by clinicians using different criteria at different time points. So there is rarely a single standardized label. And then missing data is another critical issue. Finally, um, clinical data is often longitudinal. So patients may have multiple measurements over time, and choices about how those measurements are summarized. First visit, last visit, or averages. These things change the story that the data tells. Clinical data reflects the realities of healthcare. Just different protocols, and then equipment changes, incomplete documentation. So understanding these realities is essential before trying to model outcomes across patients. So now let's see a pre-model checklist. So before data from a biological experiment is combined across studies or used for downstream modeling, there are a few things that need to be made explicit. You can think of this as a checklist, for understanding what is actually in the data you have been handed. First, trace the data lineage. So map how data moved from raw instrument to the file you are working with. Second, you need to understand, the transformations that occurred along the way. So what processing steps were applied, what parameters or thresholds were used. And then you need to look at the batch structure, where samples processed in groups, different days, different instruments. What about different sites? If so, those groupings also matter. And then clarify how labels or annotations were assigned. Who defined those things? And were they applied consistently, across the database, across the dataset, I mean. So, and then fifth, check for exclusions. Were any samples, measurements, or features removed? And then based on what? And finally, you need to identify assumptions. Every dataset carries biological and technical assumptions, many of which are easy to overlook if you are not involved in generating the data. So you need to have some kind of checklist. And then let's discuss common red flags. So where you can actually stop and investigate a little bit more. So one red flag is lack of documentation around processing steps or parameters. In experimental biology, actually, that is very common, right? Unfortunately, but it is a serious problem if you don't know how the data was processed. You don't really know, what it represents. So, yep. And then another red flag is missing metadata. That is, information about batches, dates, instruments, or experimental sites. Batch effects are real and often actually dominant in biological data. And without this context, they are very hard to diagnose. So you should also be concerned if the processing pipeline cannot be reproduced from the available information. If a data set cannot be reproduced, its results are difficult to trust, right? Labeling issues are another frequent problem. If labels were assigned by different people or using different methods without checking consistency, that variability becomes part of the data. So this matters especially for subjective classifications. So watch out and be cautious of results that look unusually clean. Biological measurements are inherently noisy, so overly smooth distributions often reflect maybe heavy filtering, selective reporting, rather than actually true signal. Large shifts in feature distributions between batches actually are another warning sign. So these patterns usually point to technical artifacts rather than actually true biological differences. So finally, maybe pay attention to missing data. So high levels of missingness in key variables without a clear explanation should raise questions about how and then why the data was lost. So when these things appear, the right response is actually to slow down, ask questions, and clarify what happened to the data before using it, for your purposes. And then now let's see why this provenance is important. I mean, first, reproducibility, right? So if you cannot trace how data was generated and transformed, you cannot reliably reproduce results. And reproducibility is fundamental to experimental science. And then interpretability. So when a model suggests that um, a compound may be toxic, let's say, or that a patient might respond to treatment. So the next question is, always why? So to answer that, you need to know whether the signal comes from underlying biology or from how the data was processed. And third, collaboration. So understanding data provenance creates a shared language between scientists and engineers. It shows that you recognize how experimental decisions shape the data and that computational methods are only one part of the drug discovery process. In some areas of ML, it is possible to treat data as a black box, but in drug discovery, that approach, it doesn't hold really. The closer a model gets to influencing real experimental or clinical decisions, the more important it becomes to understand where the data came from and what it represents. Now, let's see summary. All right, let's see what all we covered in this workshop. That is, biological data undergoes multiple layers of transformation before it ever reaches to you as a final file. Along the way, important context is often lost, simplified, or left undocumented. That is the reason, the collaboration between scientists and engineers is essential. Each perspective captures something the other cannot. So experimental understanding on one side and cross dataset integration on the other side. So as you work with the biological data, you need to learn to ask specific questions about how it was transformed, how batches were handled, how labels were assigned, and what was excluded along the way, for example. So the key, point is something like this. So before data is integrated for your downstream modeling, you need to take time to understand how it was generated and shaped. So that discipline is what separates models that reflect biology from models that reflect artifacts. Thank you.